As we continue our theme of control of enzyme behavior, let's consider another means of converting an enzyme from an inactive to an active form. Enzymes that are synthesized in an inactive nascent form to be later processed toward activity are referred to as zymogens. That is, they are in a form that may be generated as active zymes. This occurs through targeted hydrolysis of specific peptide bonds within the protein. The benefit of this method is that the protein has already been synthesized and only needs some modification to become active, so a fast response to the cell's need is possible. An excellent example is the digestive enzyme chymotrypsinogen. Hopefully that name looks familiar, since it is the zymogen form of an enzyme we've already considered, chymotrypsin. It is synthesized initially as chymotrypsinogen and stored in the pancreas for future use. It is a single polypeptide chain of 245 amino acid residues cross-linked by 5 disulfide bonds, as shown in the diagram below. When this digestive enzyme is needed, it is secreted into the small intestine, and there begins the activation process. Another enzyme, trypsin, cleaves a peptide bond from the N-terminal to produce an active form, pi-chymotrypsin. Note, as indicated in the diagram, though a peptide bond has been hydrolyzed, the peptide remains attached by means of a disulfide bond. This active form then catalyzes hydrolysis of 2 more peptide fragments to give alpha-chymotrypsin, as shown below. Note that disulfide bonds ensure that the remaining fragments are still covalently linked. Keep in mind that one molecule of pi-chymotrypsin hydrolyzes a second molecule to produce the alpha form. If you examine the figure of alpha-chymotrypsin below, you can see that there are now 3 separate polypeptide chains, even though the original form was one chain. These fragments are connected by 2 of the original 5 disulfide bonds. This is an excellent example of how changes in the primary structure may result in changes to the tertiary structure. The catalytically active form is due to this very change in tertiary structure. This is a very different method of enzyme control than that of either covalent modification or allosteric effectors, and yet it accomplishes the same goal of conformational change to the active form of the enzyme.